What if one microscopic change in a virus could bring the entire world to a standstill? Here are the 3 mutations that could unleash a global pandemic. This isn't the plot of any dystopian novel. It's a looming reality of avian influenza, specifically as far as the H5N1 strain is concerned. Today, we're talking about the possible mutations that could transform this bird flu into a global human pandemic. Let's take a look into the intricate and mysterious world of viral evolution. The lurking threat of H5N1. Since its emergence in 1996, the H5N1 avian influenza virus has been a specter on the horizon of public health, as H5N1 bird flu is mutating. And now scientists are tracking what could turn this animal virus into a changes so small they'd fit on a single strand of RNA, but so deadly they could be fatal. As we all know, bird flu, predominantly affecting birds, and its jump to humans has been rare but deadly, with a mortality rate exceeding 50%. The question that haunts virologists is, what if H5N1 mutates to spread easily among humans? And now recent studies have spotlighted there are specific mutations that could make this nightmare a reality. Mutation scenario number 1, PB2E627K. Let's start with the PB2 gene mutation, notably the E627K substitution. The PB2 protein is crucial for the virus's replication machinery. In its original form, H5N1, PB2 gene has a glutamic acid, E, at position 627. And if a mutation replacing it with lysine, K, it allows the virus to thrive in mammalian cells. Now research indicates that this single amino acid changes like giving the virus A to our cells, which enhances the virus's ability to replicate within human hosts, potentially increasing its virulence and transmission. Alarmingly, this mutation has been observed in 319 cases, where H5N1 infected humans highlights its role in cross-species adaptation. Mutation scenario number 2, hemagglutinin, HA cleavage site alterations. Next, we examine importance of mutations in the hemagglutinin, HA protein, specifically at its cleavage site. HA is the key that allows the virus to enter host cells. For H5N1, a polybasic cleavage site enables the virus to be processed by a wide range of host enzymes, facilitating systemic infections. Studies have shown that certain amino acid substitutions at this site can dramatically increase the virus's pathogenicity. And for instance, a serine or arginine substitution or the deletion of glycine at position P6 has been linked to heightened virulence in mammalian models. Such mutations could render the virus more adaptable to human hosts, raising the specter of a pandemic. Possibility number 3, reassortment with human influenza viruses. Perhaps the most insidious scenario will involve reassortment. That means the mixing of genetic material between H5N1 and human influenza viruses. This can occur when a host, such as a pig or even a human, is co-infected with both viruses, allowing segments of their genomes to shuffle. The result? A novel virus with the surface proteins of H5N1 and the internal machinery of a human-adapted flu virus. Such a hybrid could combine the lethality of H5N1 with the transmissibility of seasonal flu, a combination that could spell disaster on a global scale. Now let's take a closer look at the impact of the global spread and its implications. The spread of H5N1 has been relentless. Initially confined to Asia, it has now reached Europe, Africa, and the Americas. The clade 2.3.4.4b variant, in particular, has demonstrated an unprecedented ability to infect a broad range of species, including mammals. This expanded host range increases the opportunities for the virus to acquire the mutations we've discussed. The recent detection of H5N1 in dairy cattle in the United States and its transmission to farm workers highlight the escalating risk. Each spillover event is a roll of the dice, with the potential to produce a strain capable of human-to-human transmission. In response to these looming threats, Now global health agencies are on high alert. The World Health Organization has emphasized the critical need for vigilant surveillance of avian influenza viruses in both animal and human populations. This includes monitoring for the key mutations we've discussed. Additionally, research into preemptive vaccines is underway. Notably, experimental mRNA vaccines targeting H5N1 have shown promise in animal models, offering a glimmer of hope in our preparedness efforts. Even if we survive, the virus could finish us off. Because H5N1, creating a playground for drug-resistant MRSA, which has mortality up to 35% of infected, and flesh-eating strep, which has rising in post-flu cases. According to the CDC's 2024 report and historical reviews, bacterial pneumonia played a significant role in the high mortality rates during the 1918-19 influenza pandemic. With today's antibiotic resistance, history could repeat. The mutations we've explored today, PB2E627K, HA cleavage site alterations, and genetic reassortment, are not just theoretical concerns. They represent tangible threats that could ignite the next global health crisis. The scientific community's race against these mutations is a testament to human ingenuity and resilience. However, public awareness and support are equally vital. By understanding these risks, we can collectively advocate for policies and practices that mitigate the chances of a pandemic. The vigilance of each individual contributes to the safety of all. Thank you for joining me on this deep dive into the mutations that could transform H5N1 into a global disaster. Thanks for tuning in to Health Bulletin, where facts matter. If today's story gave you something to think about, don't keep it to yourself. Share it with others. Hit the subscribe button so you never miss an update. Drop your thoughts or questions in the comments. We read everyone. And if you've got a story or want to contribute, reach out at the email in the description. Until next time, stay curious, stay safe, and above all, stay human.